So a few times I had the following question. So people who got multiple FASTQ files per sample without having paired end data. So you know that when you have paired end data, it's normal to have two FASTQ files per sample, one with the forward reads and one with the reverse reads. Now, what if that's not the case and you still have multiple FASTQ files per sample? Well, the most likely explanation for that is that you had big libraries that don't fit one single lane. So if your experiment is so big, you have many libraries, the libraries are quite large, then it might be impossible to all fit them into one lane on the flow cell. So what is the sequence provider going to do? He's going to divide them over the multiple lanes. Now, he's not going to do the following, half of the samples in one lane and half of the samples in the other lane, because that would create a lot of variation between these two sets of samples that is not biological. So better solution here is when he fits each sample over both lanes. So he's going to divide each sample in two and put half of it on lane number 1 and half of it on lane number 2. Hence, you will get two FASTQ files per sample, because what the sequencer generates is one FASTQ file per lane. That is then demultiplexed into one FASTQ file per sample, but since you use two lanes, you will get two FASTQ files per sample. Okay, normally your sequence provider should fix this, but they don't always do this, because I get this question sometimes. So what if that is the case? You have two FASTQ files per sample and it's not paired end data. Well, a good idea is to do your quality control, so the FastQC analysis, on these files separately, because different lanes behave differently. One might be overloaded while the other one was not, for instance. So there might be issues in one lane that are not present in the other. So it makes sense to look at the files separately when you want to evaluate the quality of the reads. However, before you do the mapping, you have to combine these files again, because some mappers can really not handle that. They want to have all the reads for one sample in one file. So how can you do this? How can you merge these two FASTQ files into one? Well, there are tools for doing that, and in Galaxy, the tool that you use for that is called concatenate datasets. So here in the tools section of the user interface, when you type concatenate, here we are, concatenate datasets head to tail, this tool will allow you to merge multiple FASTQ files into one FASTQ file, which is exactly what you need before you do the mapping.